Welcome everybody to London Futurists, where we often say that the most interesting scenarios about the future aren't simply extensions of single trends, it's where there's convergence of more than one thing, and it's in that overlap that the biggest surprises, the biggest risks, and the biggest opportunities arise. I've had the pleasure over the last couple of weeks to read all the way through the book that today's speaker has written, and that's the same message I got very much in the book, that the most interesting things to do with the future of biology, the future of healthy longevity, and the future of physics in many ways, is in an overlap. It's not just a single-minded pursuit of a single line of inquiry, it is in being open to multiple insights from multiple perspectives. And I must say, I feel considerably wiser now, having read that book. Not only was I aware of interesting breakthroughs happening in laboratories around the world, that's both academic laboratories but also commercial laboratories, I was also challenged and inspired to think in broader ways about how these breakthroughs might happen with broader kinds of collaboration, not just multiple teams, but teams applying multiple disciplines. Albert Einstein is said to have expressed the view that we should strive to make things as simple as possible, but no simpler. By which he meant that if you just try to force an idea into the richness of reality, sometimes you will have too simple-minded an approach, and actually we need to be able to step back and see things with a bigger perspective. That is very much what I'm expecting is going to happen today. The speaker, Professor Sonia Contera, is a very interesting title. Is it biological physics? Physical biology?
Biological physics.
Biological physics, right. And she has been in this field for, let's say, a number of years, and has seen some ups and downs, and I'm sure she's going to be sharing some of her own experiments, her own findings, but also more widely, what is happening with this field of nanotechnology, and to what extent can we say this field is coming to life. So let's welcome Sonia Contera.
Thank you. So the title of the book, Nano Comes to Life, already points towards the first convergence that nanotechnology facilitated, which is the convergence of nanotechnology with biology. Nanotechnology is the capacity to see, interact, and fabricate matter at the nanometer scale. And from the early days of nanotechnologists like me got obsessed with biology because we are built up of nanomachines. Our proteins are the most sophisticated nanomachines that the universe has created as far as we know. And from the early days, we were interested in why and how nature created us from the nanoscale up. So, since this is talk about biology as much as it is about nanotechnology and physics, let me start with this. I'm sure you're familiar with the type or description of biology. Despite our extreme complexity and the complexity of the world around us, from plankton to bacteria, to trees, to cities and societies, in the last maybe 30, 40 years, a big part, not the whole community of biology, strive to simplify biological complexity to DNA. DNA is 2 nanometers, is a molecule 2 meters in each of your cells, and is 2 nanometers thick. So it's in the realm of nanotechnology. And this picture, I guess, is familiar to all of you because it's in the media. You usually have a more or less male hand manipulating genes, and the genes are associated with intelligence or human traits. This picture has no scientific foundation. And I will try to tell you why we arrived to this picture and how nanotechnology and many of the sciences converging right now into biology are shattering it. So, the problem with biology is that we always knew that there was a lot of stuff inside of us. This is known to all of us from the early civilization. It is not until the late 1600s, the 17th century, that Leeuwenhoek and then Robert Hooke in Oxford start creating microscopes that I start to look into biology. When Robert Hooke looked at cork, he identified little holes in the cork. This is the first time we started to see some zooming into biology, and he identified this. He didn't know what it was, and he called them cells. The reason he called them cells is because it reminded him of the little rooms where the monks were living in monasteries. They were called cells. Now, it took much longer to reach the next level of complexity, because inside of the cell, there's so much stuff and so small that we didn't have the capacity to look into it in many centuries. So the main problem is that the constituents of the cells are in the nanoscale, and light microscopes cannot see beyond 300 nanometers. That's the diffraction limit of light. It's impossible with a light microscope to see, well, until now. People are getting clever, as I tell you later, but indeed it was not possible to see. The breakthrough to see matter at the nanometer scale came with Xray diffraction, crystallography in the early 20th century. That's the moment where people realize that X-rays are not only useful for seeing through your skin and see your bones, which is the beginning of X-rays being used in medicine. Physicists have always been involved in biology and medicine, despite the overall impression that we haven't. In fact, Einstein and all the main breakthroughs that we have had in physics and the major changes we have in understanding of reality, quantum mechanics, and especially of general relativity, comes from profound reflection by physicists of how we learn, how humans learn, which is in itself a biological question. So, X-ray diffraction, people realized that the length is so short that when you throw it against a crystal, such as the salt you have in your food, crystals are arrays of atoms. There's a lot of materials on Earth that come in arrays of atoms, which the atoms are placed in perfect order, forming a perfect geometry. And if you if you throw an X-ray to that, it diffracts. And from the diffraction pattern that you get extended against a film, just a photographic film, and a lot of maths and a lot of thinking, you can figure out what is that crystal made of. That immediately got physicists obsessed with trying to make crystals of biological components so we could figure out their structures. It took a long time. So in the 1950s, it started to come in the first breakthroughs. So the guys involved that were famous at the time were Linus Pauling in the US, Crick here in Cambridge. And they were starting to get the first results. The main problem was that biological materials are not made to be made into crystals. Our proteins are swimming around in water and doing a lot of movement. They're not. So it took a long time and a lot of ingenuity to create those crystals. There was a guy in Switzerland that everybody forgets in this story that was making crystals, or at least fibers, very purified fibers of DNA, with from the sweetbreads of Swiss cows, and he sent them over to London, to Wilkins' lab in King's College, and Rosalind Franklin came to work there, and she worked really, really, really hard to try to figure out the structure of DNA. She got many pictures like this one, and from this structure, she thought, and many people thought, especially her and her student, Rosalind, that DNA had a helical structure. Like many women in science, still now, we know our findings are never taken seriously, and that we face a lot of stereotyping of the way we present our science and we do science. So Rosalind Franklin decided to repeat and repeat the experiments, analyze the experiments, until she was convinced she had enough evidence that she could say this is the structure of DNA. Maybe for better or worse, Watson and Crick got hold of her data that she shared with them for discussion. They published the paper without her. They got a Nobel Prize. She died of cancer because of doing the experiments. And I think the pictures are also quite interesting to see the two Nobel Prize winners and the hardworking woman in the lab that didn't get it. Everything reflects the gender politics of this game. So it was not just DNA. We started to get the structures of proteins. We are made of proteins. Proteins are little balls that our cells produce. So the central dogma of biology, enunciated by Mr. Crick just after they discovered the structure of DNA, is the following. Your DNA encodes all the genes of your body, and then the cell reads the information, whatever and how he reads it, he didn't elaborate. And then you produce protein, and first you produce RNA, and then from the RNA you go on to code protein, and then that's the way the cells construct our cells. So proteins are our main constituents. Our main structure is made of collagen, coating the bones also cells are stuck on collagen, but then you have amazing nanomachines like the little sensors you have in your eyes that get the light in and get all the information in, all the the pressure sensors in your fingers, temperature sensors, all the devices in your cells that allow the cells to function. We are made of nanomachines. So these guys spend a lot of time creating crystals of proteins, and at the beginning when there was no computers, this was quite hard work. One of the most prodigious person to do this type of work was Dorothy Hodgkin, which you may have heard of her, who had prodigious skills for figuring out the structures out of this. That's why she's famous and still is the only woman in the UK that have got a Nobel Prize in Science. That's also the reason why we have the Diamond facility. The UK pharmaceutical industry is quite strong, and of course there's a lot of research in this area. Crystallography has been very strong in this country. All the people I've been telling you are British. So in order to create more and more understanding of the structures of proteins, you create more and more powerful X-rays, so you don't need such big crystals. So that's one of the reasons why we have the Diamond facility. So 20th century, mainly obsession with molecules. So this is a reductionist approach to biology in which we put an enormous effort in understanding proteins. Why? Because pharmaceutical industry thinks that the way to cure disease is to identify either the protein or the gene that is faulty, and then create a drug to silence it or get rid of it. This picture totally misses the interactions between them, or how difficult it is to reach them, or how come proteins interacting together create such complexity. They break down biology to the constituents, which is the obsession of science for much of the 20th century. And they produce pictures like this, pictures in which you have incredible messes that things are related with the strings, no mechanistic models, no physics here. And a DNA which is central to the whole story, just still not realistic, because in a real cell you have 2 meters of DNA stuck in 5 microns, so the physics of the DNA or how the DNA might interact with the outside world is missing. So this is a reductionist vision of the world, a vision that is seeking simplicity in the chaos of the natural and social world. This is not just a scientist approach. Of course, science happens in the context of societies. And this idea that we could reduce humans to genes was also very useful for the social and economic context in which this biology happens. Of course, it's useful for certain political agendas, such as colonialism or even patriarchy, because we have a way to measure people, like, and create hierarchies about people. So these are complicated stories. Why scientists work the way they work? How do we get the science? We get the funding to do our sciences, and which scientists get Nobel Prizes, as I told you before with Watson and Crick. And this is important because the mythology of science is very important to how science works and the sociology of science. So, another part of this agenda is that reductionism was, since the Industrial Revolution, very useful to create tools and create technologies to dominate nature. And indeed, colonialism, in the sense that the warfare, the ships, all these are based in reductionist approaches. So humans for a long time learn how matter works, the constituents, and from that way of thinking creates a kind of technology that is always sort of top down. We think it with our head, how we put the little bits together and create a technology. And that gave us a kind of impression that humans would overcome nature. We wouldn't have to think anymore about our diseases. We will kill our proteins or our genes. In agriculture, we will create molecules. We wouldn't need anymore to think harder about fertilizers. So it was a period of human history where reductionism and technology for dominating nature really took control. The atomic bomb, again, reductionism. But things were changing, started to change in the 1980s. In the 1980s, it started to appear the microscopes that I've made a living on still now, that first allowed humans to look at matter at the atomic and nanometer scales. These microscopes were not based on light, but they were based on, if you want, touch. Interactions that have a very sharp tip with matter. Carefully controlling those interactions allowed humans for the first time to see atoms. This is a famous example for still my favorite ever microscopy image. It was done by Don Eigler in IBM in Almaden in 1990, I think, 1992. What you see at the back is a copper surface. The little balls here are atoms. On top of the atoms are electrons, but because it's a metal, if it is here, the characteristic of metals is that conduct electricity. The reason they conduct electricity is because the electrons run very freely at the surface. That's why you see kind of wavy, free, liquid-like coating of the atoms with electrons. So the Eigler, this lowered the temperature of the system and put iron atoms on it. So these little balls are single iron atoms. Beautiful. So he figured out with his tip how he could pick them up and arrange them. This is the first time in human history that we construct anything with atoms, with atomic precision. It's extremely reductionist. One by one, I make a circle. This is Lego at the nanoscale. But nature is already telling you something wonderful in this picture. You may try to be reductionist, but nature is always more interesting. You see a standing wave in the middle, a ripple. And the ripple are the electrons. The electrons here swim freely, but here they're confined by the quantum corral, as they call it. So what we see here is a standing wave emerging, and this is an important word, from the interaction of these electrons with the circle. So this looks very simple, but we already see complex behavior that cannot be explained just thinking of the constituents as soon as you deal with matter at any scale. Nature is always an entanglement. Everything is related with everything. And this is something that we did not think about for most of the 20th century. Physics did, and didn't have any other choice with the beginning of condensed matter physics. All the devices you have in your mobile phones are based on silicon, superconductivity, magnetism. Physicists started to think, what happens when the atoms come together? They share electrons. What behavior do they have? They started to create theories of complex matter. So in the 1980s, what we had about biology were cartoons. The cartoons people were figuring out of the crystal structures, but up to this moment, we could not see proteins in their real environment. So me and others, well, I got inspired actually by this. I started to think, how can we use the new tools of nanotechnology that would allow us to see atoms to study biology's nanomachine? Let me introduce you to the ATP synthase. This protein, for which these guys got a Nobel Prize in 1997, Scott Peterson in August, John Walker and Paul Boyer here in the UK, are what we call molecular motors. So this is an ATP synthase. You have them in kilograms in your body, and this protein is known, it produces ATP. You need about 60 kilograms a day of turnover of ATP for your bodies to work. It's the currency, chemical currency of life, right? How does it do it? Just by looking at it, because this guy got the crystal structure, they thought this thing probably rotates. It's probably sitting on the membranes of cells, they knew that. And it's using, this is their model, protons, charge to get a rotor rotating, which translate here, and this is what is using to catalyze a chemical reaction. Hiroyuki Noji in Tokyo Institute of Technology at the time, working with Kinoshita Sensei. I was doing his PhD, and I attached a filament to the ATP synthase and observed for the first time the rotation of the ATP synthase. So we were starting to learn about biology's nanomachines. And me and others started to use the microscopes nanotechnology were using to look at biology in their medium. So this is a real molecule of DNA, not something of a cartoon coming from Xray structure, structured from bacteria. It has a lot of tension, it has a lot of twisting, it has a lot of physics. In some areas you can see the double helix, in some areas you don't see the double helix. And crucially, our new microscopes allow us to see for the first time dynamics. We start to see how proteins move. Now movement is key, because at the nanoscale, you're so small when you're in warm water, and our body takes a lot of effort to keep it warm, 37 degrees. You're so small that the water molecules moving around move you. When they hit you, you feel it. So what proteins do is to have a mechanical design, a bit like a windmill, that when the water molecules hits them, they can produce movement. This is what evolution has produced. This is the ATP synthase looked from the top, and these are the movements of the ATP synthase when it's producing ATP. This beautiful thing down here is myosin, again, one of the molecules you have in your body in biggest quantity. This is myosin walking on a track. The track is acting. Your cells are full of cables, full of mechanical cables that communicate the inside and the outside of the cell. And very often these myosins are walking on the cables. The reason they can walk is because of their design. They have 2 feet. The feet stick electrostatically to the track, and then as water hits them, they move forward. Sometimes they use also ATP to make the movement more easy. So this is the reason why biology happens at the nanometer scale. When you're so small, a polymer folds into a shape, and that shape, over millions of years of evolution, has learned to encode information that is enough to use the energy of the surrounding to produce a mechanical movement. That movement is usually coupled with chemistry. So chemistry in biology doesn't happen like in liquid, like the chemistry humans do. Chemistry is hardly harnessed by mechanics, by structure. So when a protein like this one grabs ATP, grabs it, and because it twists, it sticks and twists, bends the molecule, and the electrons jump. So you're facilitating, that's how you make a chemical reaction. That's catalysis. That's the most clever catalysis you can do. And that's how biology works. In the 1980s, Eric Dressler, who wrote the famous book Engines of Creation, which created a lot of debate, is controversial. It misses part of the science. Because one of the interesting things we see here is that in order for these machines to work perfectly, they need to be in the perfect environment. One key characteristic of nanotechnology, when you're so small, if you even change the concentration of the pH or anything in your environment, you don't work in the same way, because you feel everything. That's why the nanotechnology goo that he was predicting is really unlikely, because as soon as you leave this here, it will disappear. Biology took a long time to create enough stability on Earth for these nanomachines to be stable. The beginnings of life, the Earth is about 5 billion years old. Life on Earth emerged about 4.5 billion years ago. And it took a long time to create stable conditions on Earth, so life was stable. There was mass extinction after mass extinction, because we didn't have a stable pH in our seas. We didn't have a stable soil concentration. Something I've been reading recently, for example, plankton appear and stabilize the pH of water. That's why we are now in big danger at the moment. So one key of nanotech that is a big problem for non-biological nanotech, the nanotech we're trying to make for our devices, is that any change in your environment, you feel it. Because you're so small that your surface is bigger than your bulk. You're all surface, you're all entanglement with the medium, which is, again, a key characteristic of biology. These structures emerge from the universe from the entangled with the environment. They're not separate. They're not things you can change and cut and paste like the DNA of the pictures. Now, in the 1980s, people start to make also fullerenes, nanoparticles, nanowires. Chemists and material scientists learn how to build things at the nanoscale. So we start to be in a scenario that suddenly humans start to reach the nanometer scale, and we want to do many things. We are very reductionist at the time in history, and of course we think we can break every single problem with our approach of we just get particles or small bits and we will solve them. That was the beginning of nanomedicine. One of the main problems, for example, of chemotherapy is that when you have a tumor, in order to kill or try to kill, or at least kill a few cells at the surface of the tumor, you need to put very high concentrations of drugs. So the drugs reach the tumor enough concentration to kill it. That means it's killing the rest of your body, as you know, and chemotherapy. If you had to go with chemotherapy yourself or through someone, it's one of the hardest ordeals you can get yourself and your family through. Now, in the 1980s, again, Maeda in Japan finds out that nanoparticles stick or accumulate in tumors. The reason is that because tumors create these veins around them, that they're not very good, they have holes, they have fenestration. The nanoparticles can go in, and in principle they should facilitate the delivery of drugs. Since then, and now, people have been working really, really hard, creating the more incredible and clever nanoparticles that will reach a tumor, stick to the tumor, and either deliver drugs or use magnetic fields to hit them out and burst them, or use light that will be absorbed by a particle and kill the tumor. Unfortunately, very, very few of these nanoparticles become available for treatment. In some ways, nanoparticles reproduce the methods of pharmacology and reproduce the results. Pharmacology in the last 20 years has not produced many drugs that are good, apart from, we had these wonder years of drug discovery with the antibiotics, the statins, the chemotherapy drugs, but there's big stagnation. Because this method of trial and error to produce drugs, the drugs are produced not by, don't think that people sophisticatedly, very, very rarely, still now, people find drugs by designing molecules that attach to proteins. In some cases they do, but mostly they just try the molecules they can make into cells in plates and try to see if there's a response. And somehow these guys did the same. They just were making nanoparticles and throwing them to cells in plates and mice in labs to see if they could do something. They cure mice in labs, but they don't cure humans. And the reason is because they always, as usual, we are trying to overstep biology's complication, like we do with climate, like we do with everything else. We think we can just go and fix, and we don't want to face this is a complex problem. Actually, talk about Einstein. When Einstein had the theory of relativity at the beginning, he realized he did very complex maths, and he didn't like it. He thought, God cannot be creating the universe in such a complex way. But he gave in. He said, yes, it is very complex. I had to speak to mathematicians. Relativity is complicated. Somehow we're learning now in biology. I will tell you later, but it's taken a long time. Now, another area that is notorious for its failures is biosensing. And this has been notoriously famous because of Theranos, that you may have seen the movie or heard about her. So there was a very bright and obviously very clever woman in the US, very young, that convinced a lot of very famous, important, and rich people to give her money to create a company that will be able to detect any disease just with a drop of blood. She was picking up from a long story that has been going on in labs since I remember, since the 1990s. We thought we could make devices and just very simply we could put there some antibodies and we pick up everything in the blood. But of course you put this in blood and everything gets coated, a mess. We have so many constituents, it doesn't work. Theranos didn't work either. They couldn't manage to break this problem. And actually, after many, many years, the breakthroughs are finally coming. And just a couple of weeks ago, I think Hitachi announced for the first time a device that with a very quite sophisticated, after many years of research and a lot of people, that it start to be able to detect a lot of chemicals in blood. I think it's also interesting who develops these complex technologies. That's for another discussion. And I would argue that startups alone will not make the breakthroughs. Hitachi and many Japanese companies, the system is very different, like Samsung does, employs a lot of scientists, has a lot of research programs that they put in collaboration with universities. They reinvest a lot of the revenue back into very basic research, and they are able to put programs for many years until they reach a product. It's very difficult with the model of startups that we have now in the West to produce these results. And a startup is very difficult and relies on luck, and starts with a very small group of people, and you have the venture capital. It's very complex to develop complex technology just with venture capital, but that's another discussion. Biosensing is getting better. Samsung and some US groups are reporting for the first time patches that are able to measure glucose in the blood, for example, and slowly release insulin. Still, it's not as good as your pancreas, but we're getting better. Progress is happening. Another thing in this field, as in many other fields, first comes the hype to get the funding, to get the egos, to get the big labs, to get the politics going. And after the hype goes, progress continuously happens when there's less egos and there's better teams, more multidisciplinarity, and things start to come through later. And in places that there were not the places where it started at the beginning, usually, which is also interesting. In the 1990s, 80s, people also started to think of biology in a different way. Again, this is very reductionist. So DNA is made of 4 bases, and 2 bases, one of the bases only stick to the other, so you can create a kind of Lego material. And some people, as what's his name? Nathan Simon, who was going to lose his tenure track in New York, saw this picture. He didn't know what to do with his DNA. He was not getting good results with his DNA crystals. And he says he saw this picture. This is by Xray. Xray's brother was a crystallographer. One of these guys that was interested in Xray diffraction for getting the structure of atoms. And he became really interested in crystallography. And he started drawing arrays of just drawings in crystal structures. Seaman, who was a crystallographer, saw this and recognized it immediately. So there's interesting interactions between art and culture and the sciences. And he saw the 4 bits here. So, oh wow, if I manage to create a DNA shape with 4 ends that is complementary to another 4 ends, maybe I can start building with DNA. He succeeded. He and his wonderful, many of them Chinese students over the years have created the field of DNA nanotechnology. Now it's an established field. And they first started creating little structures like arrays. You can see them with atomic force microscopes. These microscopes I told you at the beginning, because they're very small. And a wonderful breakthrough came when a guy at Caltech created DNA origami. So this is the first time people can program in the computer a shape and build it by itself precisely with atomic precision without having to build one by one. What did he do? He got a long DNA strand from a virus, natural virus, studied it, and then created staples. The staples will bind in specific places, and he made a computer program that would say, if I design these staples in this way, I could make any shape. So that's the program, and that's the reality. He could make anything out of DNA. This is very cute, and it's a proof of principle. We still don't really know what to do with it. And now we can do that in 3D. So people now create any structure you can think of. It's become possible to create anything with atomic precision at the nanoscale with DNA. People are putting a lot of effort in trying to get bigger, because of course we are built of the nanoscale building blocks, but we breach all the scales. And now people are making teddy bears that are a little bit bigger and all sorts of the structures. my own contribution to the field was to create some handles that we could, with Japanese collaborators, that would pull a stick to cells with the hope of pulling from cells. But as many applications of DNA nanotechnology, you create something very small, but how do you interact with it? One of the main problems of nanotech is when you make something very small, what do you do to make it work? How do you see it? How do you make it functional? So we're still there in the field. I just collaborate with people doing DNA nanotechnology. I'm not a DNA nanotechnologist. People are getting to do more interesting things, such as using DNA as building molecules. Molecules are notoriously very complex to build. That's why organic chemistry is difficult, and that's why we have a very limited amount of drugs we can use in our body. Our body builds complex molecules not by throwing things into a test tube. I showed you these wonderful nanomachines. We have nanomachines such as a ribosome that grabs a molecule, grabs another molecule, and sticks them together. Because otherwise it would be too costly, too slow. Life could not happen. So people are trying to make primitive versions of these out of combinations of proteins and DNA. Still work in progress. I was very happy to see the other day in the Times that the Times is talking about DNA nanotechnology. These guys created a star of DNA that traps a virus. It's actually very beautiful because we're not looking now at drugs that just bind a specific protein. We're creating bigger stuff, things that goes a bit like the body does. Some beautiful thing you may know or not is the immune system of our body, our immune cells, when we have sepsis or a big infection. They burst themselves, or they get their DNA and get it out of the cell and burst it to create a DNA trap that engulfs bacteria and gets rid of them. So what we are learning really is something that evolution has done from the beginning. DNA was not only genetic material, it's also a constructor material, and our body uses it a lot, even to create nets to defend itself from invaders. So this is a moment where also David Baker starts to, and many others, to crack a fundamental problem in biology for many, many, many years. Proteins are made of a long string, polypeptide chain. And when you put it in water, it falls in the right shape. And you've seen these amazing shapes. They're walking ones, they're rotating. I mean, they're amazing. And for years, people just tried to break the problem of protein folding by brute force. So basically, right now, to know a protein structure, you need to purify it, to make a crystal, to take it to a synchrotron. So what would happen if we just know the genes, could just predict the structure? We would save a lot of money. We couldn't. We couldn't, even though people put an enormous amount of effort, crowdsourcing, collaborations, using everybody's computers to do the calculations. It's still very difficult. So these guys have a competition every year to see if they're getting better. So all the protein prediction groups go through this competition and they try to see, they give them a sequence and see if you can fold it. And one year, 2016, was it earlier, these guys cracked it. For the first time ever, these guys managed to get correct protein structure. And they did something that, in my opinion, is revolutionary. They did not try to just fold it by brute force of physics, just getting the constituents and try to see how they come together. The only way they succeeded is when their algorithm took into account the evolutionary history of the protein. So they look at all the ancestors of these proteins across the species, and they started to look at how the genes would communicate with each other. And from there, they started to figure out where were the bits of the protein that would come together probably during folding. So this is a very different way of doing simulations. And it's pointing already that we are leaving the reductionist approach. We don't only go for brute force. We look at where we come from. We look at the evolutionary history of Earth. And a beautiful thing of this field is as soon as they managed to predict proteins, they started to produce proteins that don't exist in nature. So what do? They went to their computers, they designed proteins without looking at the proteins that live in the natural world. Then they went back, they produced a piece of DNA that encoded for that protein, went back to a cell and got a cell to produce a protein. The proteins, now again, this is a highly non-reductionist approach to technology. We fabricate in the computer, we design in the computer, but then we take back to the cell, the gene, so the complexity of nature creates our building materials. We're not anymore creating technology, this is synthetic biology or part of synthetic biology if you want. We're not creating technology apart from nature, we start to merge with nature. And this is, for me, a big breakthrough. And it's a breakthrough that is happening in other fields, as I tell you now. One beautiful piece of work of this group is that they created a virus that doesn't exist in nature. So they designed the proteins that don't exist, and then they throw them into an E. coli culture, and together with it, they put the pieces of DNA, RNA that encode for the protein. They put it together there. It assembled, and it started to evolve. So in a very complex strategy that involves computers, protein, building by proteins, cell cultures, we are radically moving away from the way we've been constructing. We can create atomically precise structures that they can be built by biological organisms and they can be deployed in medical contexts. This is a very different way of doing medicine. And this is nanotech. This is building proteins, which is maybe the original nanobots' dreams were not doable because we will never, I think, be able to fabricate these structures with our reductionist approach, with a metal nanobot. What we are learning is that if we want to build clever, we need to understand and merge with nature. Now, physicists like me, because we're obsessed with big questions, for a physicist is always the cosmos, we start to think, okay, now we need to rethink everything. We come into a point where we need to understand nature, where we come from, in a radical new way. So for people like me, I don't know if this is going to work because it's not my computer. Yeah, that's DNA. Look with a high speed atomic force microscope. We start to rethink how life arrived on Earth at the nanoscale. So why life emerged? Why every single living organism needs and encodes so much information? And why all living matter has the capacity of computing solutions of survival, of intelligence. Now, revisiting physics, we know that entropy grows. That's one of the things we learn. Everybody knows this. Yet we are made of the opposite. Life emerged on Earth not for creating more entropy, but creating complicated structures. We are made of removing chaos. We are made of creating a structure out of the chaos. How does this happen? Because polymers such as DNA, when you are in water, are able to wriggle. Wriggle is a very important thing because when the water hits you, you start wriggling. You start to dissipate energy into the environment. You start to move, and you start to be able to coil depending on the forces you have around you. So those start to create a shape. If the shape endures with time, you're reducing, the shapes. You're creating a structure. It's because you're encoding information about the environment. Why? The information to survive in that environment. And if you move forward and you become more and more complicated, what I'm telling you is that the universe creates computation intelligence in order to survive, in order to create living organisms. So we are in a very different scenario right now. We are starting to understand and acknowledge that we emerge from a complex entanglement with nature. We're not as superior as we thought to nature, as the climate change emergency tells us every day. And we need to reassess, I think, at every level, our reductionist approaches and the way we're going to move forward with technology. Another piece of information that came around the time when all these things were happening was the end of the Human Genome Project, 2003. The Human Genome Project was all about, we are going to know what humans are because we're going to tell you all what you are. You run the machine, you will know all your genes, that's all you need to know. If there's something faulty there, you will get a disease. They were totally forgetting that in order to get the disease, you need to interact with the environment where this DNA is interacting. But no, a very reductionist approach. And the big surprise came. We have over 150,000 proteins in our body. So these guys, linear as they were, they thought we would have 150,000 genes. One gene, one protein, of course. No, we have only 20,000 genes, like a little worm or like very primitive organisms. So that tells you that we are not just a genetic, a sort of biochemical or algorithm written in genes. We have 2 meters of DNA inside the cell, 5 microns inside the nucleus. And out of these 2 meters, around 7% is genes. The rest is a structure, things we still don't know. The rest is what evolution has created, so that DNA fills the environment. Things happen to this DNA. The DNA combines the bits just by structure, just by folding, which is the wonderfulness. Structure and movement and interaction with the universe around it. A universe that evolution has created. Multicellular organisms only arrive on Earth very early, very late in the history of the life on Earth, because it took Earth a long time to create complex organisms. And that DNA shape contains all that history, the history that took life on Earth to interact with the environment and create the cutting and pasting of the gene that is producing us. So it's a much more wonderful picture in which the entanglement with the universe and the history of life on Earth gives rise to our behavior in ways we don't understand, and they are not deterministic. As most of us know, there's no way to know how intelligent you are, regardless of how you measure it. Right, so it was not just genes. People start to be able to see inside cells, to see nanostructures inside cells. Mathematicians, engineers started to move into biology. We started to try to make sense of biology's complexity. This is not going to work, but this will. I'm going to show you a beautiful movie. This is Drosophila, a fly, an embryo. And just seeing this movie, you realize that we are much more than genes. So when you play it, you will see that looking from one side and looking for the other side. All these cells look the same, but times go forward, you will see the cells start changing their behavior, creating patterns in extreme complexity. But still, you will find waves, you will find patterns. And this is what mathematical biologists and physicists are trying to figure out. So that cells always shaking, always moving. Oh, we're dead there. There you go. Hey, come on. And you start to see cells changing their structures, differentiating, producing the different proteins. We're starting to learn that all this happens by not only chemical communication, but key to all this is electrical communication and mechanical communication between cells. This is the way organisms are made. This is much more wonderful than genes. Right. What we are doing, and as I tell in the book, is embracing at last biology's complexity. So we're trying to look at all the hierarchy of life. We're getting obsessed with the brain, we're getting engrossed with all the building blocks, and we're striving to build a more complete picture of humans. But it's not only been biophysicists and biologists and mathematicians. There's been other worlds that have been abandoning reductionism and embracing biological complexity. As you know, people were trying for a long time to be good at translating with computers or identifying faces, but it took a long time. The algorithms were very crap and they could not really detect things until people started to be less reductionist. They didn't want to control the structure of the algorithm so much, and they started to mimic the layer structure of the brain in the algorithm. That's the deep neural networks. We don't know why they work, but they work. So we're learning again something very profound. There's some very deep relation between the way evolution created our brain in layer structures and the way we learn. A structure, shape, is fundamental for intelligence. And some physicists are explaining this because, of course, of the rules and the ways the universe is built. So we emerge from the universe with the capacity to survive because we can interpret it. And this has been encoded not just in genetic algorithms, it's encoded in our shape. Our shape is the one that translates all the signals around us and creates the computation. And the brain, in ways we still don't know, is connected to the rest of the body, mimicking somehow the structure that allows us to interpret the universe. People are also looking at how simple organisms compute. It's a nice example of a slime mold that is becoming very popular. Now the zoos in Paris have even a special exhibition, it's called Le Blob, about the computer powers of a slime mold, which is great. It's coming to the general public. I'm very happy about this. So what you see here is a slime mold. Unfortunately, the video doesn't play. Reproducing the map of the underground in Tokyo. That's the map in Tokyo. What do they do to get this? They put food for the slime mold in the train stations, and they let the slime mold to grow. And because it minimizes the path, and they give it a bit of shape to the places it is, so it has a bit of the same constraints that you have, geographical constraints in Tokyo, you end up with a map of Tokyo. If you want to do this with a top-down algorithm, it would take you a very long time. This minimization of places is very difficult to compute. People are thinking of just using living organisms to do the computations for you. So we are abandoning reductionism, and we are combining our top-down approaches and embracing nature in a way we've never done before. This is another beautiful experiment. This was done by Jim Jimsevsky, also one of the pioneers of scanning probe microscopy, and proved something very wonderful. So what you see here, this is sea of nanowires, I think they're of silver, and he created links, lots of links between them. So he created something called criticality. So basically you have a lot of interconnections with the wire, and it started feeding electrical signals on one side that corresponded to the traffic of Los Angeles. Because you're so small at the nanoscale and so interconnected, as the current goes through you, remember our DNA filling the world, these cables fill the world. As the electrical current goes through you, the structure of this changes. And they did it for a while. And after a while, it started to produce the patterns of the traffic in Los Angeles in the other side. What this is telling you is that the universe encodes in the structure of nature and the complex interconnection of matter, the capacity of computing any matter, organic or not. We're starting to learn the roots of intelligence. In medicine, the biggest breakthroughs cannot come from pharmacology, have not come from nanomedicine, but from immunotherapy. We don't use, so we see in the press, immunotherapy is starting finally to clear some cancers. Why? Because we're not using just one protein, one gene. We use cells, the incredible computer power that the cell has. And the immune system, human or any immune system, has evolved over billions of years to do that job. It grabs a bacteria, twists it, tastes it, they get all the chemical information, they get mechanical information, identify it, crack it. Now the challenge now, and people have realized, you can get the immune system to identify your cancer cells. We're still early days, but clears lots of fluid cancers, if you want, the ones that don't make tumors. One of the main problems, and that's the problem I work with, is that tumors are so clever that they prevent immune cells to go through. And this is maybe where, and it's the problem I'm working, where we can help the immune system. We train the immune system to find cancer cells, and then we use physics or chemistry to facilitate the job of the immune system. Again, not working separate from nature, embrace nature power and use our approaches to combine with them, combine the way we do technology, learn from nature how you do things, and do it together. In a way, I think people don't like it because it's giving up something we like. that our powers of technology dominate nature, are superior to it. We want to know how it works in a very human way. We want to be able to see the bits because we want to know how the cell is doing it. And there's a sense maybe of failure when you just use a cell to do it for you. And I think maybe this is something that may change or not. This is the challenge right now. Can we learn these things or no? How do we learn? But it's the only way forward. I mean, if you clear your cancer, you don't care. And if we save the planet doing better technology, we won't care either. So we're learning to be humbler, in my opinion. And maybe this is another of the reasons we're learning. And nanotech works in immunotherapy. Yay, it didn't work killing cancers by itself. But nanoparticles accumulate in the spleen, and the spleen is full of immune cells. So you can use the nanoparticles to give the information to the immune cells to recognize cancer. Or you can produce materials such as these ones that are nanoporous that continuously release molecules, information to the immune system to keep targeting cells. So these combinations of material science, computer, nanoparticles, knowledge about biology is bringing the breakthrough. And I finish with tissue engineering, which is another of my favorite fields. So apart from curing cancers and horrible infections, people always want to regenerate body parts. It's very difficult to go through life in one piece. Most of us lose some bits in the way. I do have scars. So we always wanted to regenerate ourselves. And how does the body, or in this, how do frogs regenerate themselves? Many animals seem to be able to do it. And we're learning how we do it. So wonderful work by Michael Levin in the US is rediscovering work that was done early in the 20th century and reassessing the role of the connection, the electrical connections between the rest and the rest of the body to be able to regenerate. The brain sends electrical information all over the body and the cells respond to it. When you have a wound, if you put a voltmeter across the wound, there's a voltage drop. The cells create a voltage. If you put a voltmeter between your head and your feet, there's a voltage drop. We're polarized. We're full of electric fields. Totally overlooked in the last 20 years. We're just looking at genes and proteins. And now we're rediscovering mechanics, electricity, how the whole body is composed. If you want, we're going back to traditional medicines. Most traditional medicines, especially Chinese medicine, is very Chinese in the audience, or is very concerned with the physics of the body and how the whole body reconnects. This is an interesting connection because a lot of this work is done in Korea and China and Japan. So I think we're going to see a lot of interesting things, culturally different ways of doing medicine in the future. So one beautiful experiment that was done was that if you get a stem cells where we come from originally, you come from a ball of cells that they're all the same, and you put them in substrates of different hardness. If you put the cell on something as hard as bone or something as soft as brain and you let them, they stick, they pull, the DNA feels it like a mechanostat and start churning protein. And the cell that sits on something as hard as bone turns into bone cell. And the cell that sits on something as soft as brain turns into something similar to a neuron. So in our evolutionary history, there's not only chemistry, there's also physics, there's also mechanics. Indeed, we move around. We are mainly mechanical. And mechanics is important. So what we are learning or trying to do is to reproduce the mechanical environment of cells and electrical environment of cells to create new tissues, such as cartilage, which is a big problem for many people. Can we make cartilage? Not yet. But there are successes in using the materials of the body to create body parts. So instead of doing artificial materials that you can sit cells on, you get, for example, the trachea of a donor and remove the cells of the donor once it's died and removed. So then you reimplant the cells of the patient that is going to receive this organ. So what you do is and then over the time, the cells sit there, feel the right structure, the right mechanics, the right environment, and those cells start to produce protein, start to regenerate the structure, and eventually you make it yours. So this is great, and it works with a few organs, such as tracheas and simple things. We're getting better also at creating patches that reconnect spinal cord injuries. So nanomaterials, polymers that are electrically conductive that help neurons to reconnect. These ideas are implemented these days on 3D printers. So you get a 3D printer printing tissues, printing cells, and printing tissues, hoping that we can start to reproduce the environments of cells. But most organs don't like this, are this size. We're not able to create big hearts or anything like that. At most, you create bladders and simple structures, tracheas, but still not very complicated tissues. But what they're very useful is to understand how tissues are built and how we can use them to get information about tissues. So instead of looking at biology and living systems, we're learning by making biology. And these usually are complicated collaborations of engineers, biologists, physicists, mathematicians. We're all obsessed with how we create tissues. And the good news is that for many of us thought that these things would be useful for testing drugs. Instead of doing drug testing on animals, on other humans, human, which is this horrible thing that drugs have developed by men, for men. Did you know that most research is done on male rats? And that's one of the reasons that most painkillers, when you really need painkillers, don't work in women. And if you put this together with they don't take into account our diseases, and that's endometriosis and things like that, makes you feel very bad. So hopefully we can print some female tissues and start to do some drug testing there. And just last week, like 2 weeks ago, I saw this. I think this the first time a liver chip identifies drug toxicities of human, rat, and dog models. So we get starting to get there. These little chips might be useful, at least in the first stages of drug testing, before you go into animals and humans. So, what I think for the future of medicine will be something like this. We will get better at biosensing and collecting real-time data. We will use electric devices, optic devices, combinations of nanoparticles, polymers, and cells. We will merge much more with nature. We will create biomimetic materials, and we will merge things with each other. We will get better mathematical modeling. We will get a lot of data, and it won't just be machine learning and big data. That won't work. We need good models of what actually the mechanisms going before we deploy just machine learning, which is again something is happening in medicine right now. The reductionists are still there. The reductionists are trying to get every single protein, every single gene, everything you can get information of the body, and just by brute force of machine learning, trying to get the patterns of how they interact with each other. This is like looking at the stars for over a long time, of all the stars you can see of the universe, and trying to make sense with each other without forbidding us specifically to take into account gravity. One of the wonderful and most mysterious capacities of humans is the capacity to model the world with mathematics. It's a mystery, I agree, and this is why biologists don't like it, and this is why it's the job of the physicists, been since Galileo, really. When you look at stuff and moves, miraculously, we can understand it, and we can make mechanistic models of it. This is physics. This is what we do. This is what Einstein was doing, sitting at the patent office. How do I learn? And what do I learn? And just from the experimental data he got, he came out with a really freakish theory of general relativity. And it works. Because there's something magical in the human brain that we are able to understand, not just in a reductionist way of understanding. There's something very deep about human intuition. And a lot of work of physicists and mathematicians is to create correct intuitions. When we call someone you're a good physicist, it's because it's a very hard skill, and that's why physics has all these myths. You train yourself to feel I feel it, even in my stomach, when I have a problem. You feel the problem in front of you, and then you say, it will move that way. Or you apply that equation. I think our machine learning is very far from that. And that's why I think we will need clever combinations of what I call smart mathematical modeling, which will involve our innate capacity to do physics and intuit nature, to do maths, and then, of course, artificial intelligence and simulation to make sense of the law of data. Now, will artificial intelligence be able to produce maths? Probably yes. We're still trying to know how do we do it. And somehow, I think, as I said before, we're getting really close to the dream of traditional medicines, because that's what they do. If you see in Chinese traditional medicine, something Koreans really love, the pulse takers, right? So you have people that train themselves over many, many years and many, many people just to sit and get the pulse of people. And just by filling in extremely good machine learning there, they tell, oh, there's something wrong with you, right? So this is in a way what we're trying to do. We're trying to get sensors that we put in the sticker in our thing, and when we're going to get a very clever mathematical model that substitute the Chinese medicine doctor and tell us there's something wrong with you, which I find a beautiful interface, especially because many of these technologies are developed in the East. I studied in Japan for a long time. Yeah, so I want to maybe finish with this, because it's a beautiful example of the technologies that are coming. What I tell in the lab, we're moving towards transmaterial futures. And what are they? In the book, I talk about that. So we learn from biology using artificial materials. And then we learn from biology how to make artificial materials. And then we start merging them to create biology. So Kevin Kidd-Packard at Harvard Swiss Institute, which is one of the leading places in this, created this beautiful robotic string ray, which is based on all of the above. So it's made of plastic and has circuits in it and is simulated and has heart cells of rat. Not that they killed a rat, probably they were cell culture over many years. And this thing can swim because you've been designing with your computer the mechanical properties of the thing and the beating of the heart cells, and it's able to follow light. I think this is a nice example of the technologies that will come. Some bits we will design, some bits we will just use nature. And because I'm going very long, I'll tell you a bit of what I do. I already told you that I work in the physics of pancreatic tumors. Pancreatic tumors kill you with physics because they create such a tight mesh of polymers around them. This is a polymer physics problem, soft matter physics, one of my expertise, that nothing goes through. Drugs don't go through. Immune cells don't go through. I'm trying to understand how do they know they need to make it, how the cells communicate with each other to know how tight they need to be. This is what is I think it's a hard problem of synchronization. This is something we can tackle with complexity theories. One of the beautiful things of mathematics is that we are able to deal with very complex systems, and we are able to extract patterns from things like coordination. I also work in mechanoelectrical coupling in neurons. If you have Parkinson or epileptic fits, in some cases, if you focus an ultrasound in your brain, it stops the Parkinson, the shaking, stops the epileptic fit. We don't know why. It's because the brain is not just electrical. Everything that is electrical in the brain always is coupled with a mechanical counterpart. So you can stimulate the brain with electric fields, but you can also stimulate the brain with mechanical signals. So I'm looking at that. And I also work in cryopreservation. I'm interested in how come some frogs are able to freeze themselves during winter and come back to life in spring. And if we can do that not only with frogs, but with cells and all sorts of constructs that we would like to preserve in this world of global warming, might be a useful thing to do. And I just finish with an example that is not, I work with plants. Plants of intelligence that doesn't have a brain. Plants stay put, and they, I don't know if the movie will work since I transfer. Yeah, they can't move with things happening around them, and they don't have a brain. So all the information and the computer power they need to survive is in their structure. And I'm very interested in understanding that, what we call growth, form, and life. That's a little plant growing. So I'm trying to understand how it creates its shape. So I come with my atomic force microscope, I sit here, and I try to feel the mechanical behavior of the material, a bit like a mechanical engineer will do, but at the nanoscale in living plants, and create maps of how the mechanical design of the plant, this is the leaves, they make these beautiful cells with different patterns, give rise to plants. The plant grows by something called turgor pressure. So the cells of the plant have cellulose nanowires. We are always built of nanostructures that when the turgor pressure comes, expands and start growing, the cell behaves and feels it and produces more or less cellulose and has pectin. That's what we make jam from. So that's the jammy in between it. So you create a hard structure and a softer structure at the same time. So you can create shape without breaking. When the wind comes, you don't break. You can grow harder. Incredible feedback mechanisms. This is built by evolution over millions of years. The oldest organisms living on Earth are plants. So some time ago, a good friend of mine, which is a sculptor, asked me if he could, I don't know what he wanted. He asked me, he wanted graphene to be, he makes these steel sculptures. These ones are from a plant, from a park, that he put them in a park in Japan. And they're made of steel. Steel has these wonderful mechanical properties for building because between the iron atoms of the crystal, you have some carbon atoms. That makes it very hard and you can build the structures. I said, I want graphene. I said, but graphene is useless for this, nanoparticles. It's the first time I confronted the problem of constructing big with nanotech. And all really we can do, and this is what you have now is fiberglass, composites. You have nanowires made in a mesh, in a sort of polymeric solution, but they can't make anything interesting. One interesting thing I found is that because his sculptures, I think, are in a park, so they have dimensions of plants, which I like. So I started thinking about wood as a construction material of the future. Other people have done it before. Wood is amazing. Has this nanostructure, these tubes, and the tubes then get lignin in, natural resin, and construct all sorts of mechanical properties. There's a group in the US that have managed to, for example, by removing the lignin and just aligning all the fibers, to create a material that has better mechanical properties than steel, just of wood. And the key is, you don't have the cellulose by itself, the building blocks. Use nature. The nanowires are aligned. We can't possibly do that. And then they put them together. They just press it. They press the cellulose. And the material is harder than steel. There is a company in Japan now, I don't remember which one, that's trying to make a car out of this, which is great news, because we need better constructing materials. And it got me reflecting on that. And my contribution has been, I want to make transparent wood. So a friend of the sculpture, actually, who is an architect, who works for one of the directors of Amanda Levett's architects, the ones that did the extension of the Victorian Armament Museum, asked me for an idea, because they're going to the Venice Biennale of Architecture, and say to them, let's make transparent wood. Again, use nature to create something beautiful that doesn't exist. Remove the lignin, substitute it by another resin, so you can control the mechanical properties and the thermal properties of the material and how translucent it is. And if you want to see my last invention, doesn't work, my last invention, is here. Let's see if they play. Yeah, that's a magnet, that's a piece of wood. Yeah, I'm making magnetic wood. Which I hope I can do something useful. As you see, it just came out of the pot. I've been trying to grow secret magnetic materials inside of it. So, this is the end. What I'm trying to show you is that, in my opinion, nanotechnology didn't bring the engines of creation, but gave us maybe something better. Brought people from other disciplines to biology that were not subject to the politics of the genes and the proteins and the pharmaceutical companies. When I arrived to biology, I had no clue about the mess of the pharmas, or the genes, or the genetics politics. I just want to know how life worked. And like me, many engineers did, and that created radical change. We look at biology with other eyes. we bring our mathematics, our physics, and indeed our willingness to work with biologists to crack fundamental problems of the 21st century, not only in medicine, but in technology, in construction materials, in electronics. And the most beautiful thing is that I think we're changing the paradigm. We are not anymore, hopefully, just thinking about how we dominate nature with our brute force approaches and our top-down approaches, but trying to learn how we emerge from the universe, how we're entangled with the universe, and how we can use that to create a radical new technology that hopefully bring us to the survival of our species. So I finish with my friend the sculpture again, because I think it doesn't only bring us to the merge of biological engineering and physical and mathematical sciences in complexity, but I think it redefines our relationship with culture, with the humanities and the arts. Since the beginning, we've been obsessed with things like intuition, which I mentioned in physics, creativity, emotion, and the role of emotion in learning, right? I only learn when I'm emotionally engaged. Why? And so far, we've been making these questions with arts. Sculpture, abstract sculpture is particularly good for this, because in sculpture you have matter, you have shape, you have emotion, and you have intuition. And I think in the 21st century, hopefully from now on, we're going to start to re-engage with these questions from all the type of science I've been telling you before. How can artificial intelligence develop intuition? How does it mean for how we do technology in the future? Okay, so I finish here. Thank you for your attention.
I think that's one of the least reductionist talks I've ever heard at London Futurists.
Yes.
Let me ask you about tissue engineering. And as you mentioned, those of us who are getting older occasionally notice bits of our bodies not working so well. Sadly, many people's hearts get heart disease and their hearts don't work so well. Is there a possible future that you foresee in which we will be able to use some of the methods you're describing to give us replacement hearts?
Well, I'm working on it at the moment. One of the problems with hearts is that when we get older, we all have arrhythmias. Why we have that? So the heart, I've started to work with these wonderful scientists in Oxford working in heart, has these cells that are very elastic, amazing stuff. I'm using them in the lab. They connect with each other, almost like a Tetris. It's really beautiful. It's like beautiful mechanical design. So they need to beat at the same time. Tock, 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 right? And it's not just electrical, the beat is mechanical. So you have an electrical signal and a mechanical signal. As you get older, these cells start secreting collagen. Collagen and fibers are the main problems that we have. Collagen is usually behind immune diseases also. So as soon as you get fibers out of place, things don't match mechanically. So the synchronicity goes. So one of the works I'm trying to do now, trying to convince people to work on, is that nobody actually works in these problems of how mechanics is important for synchronizing the heart. So I'm trying to put together a team of mathematicians, physicists of complexity, physicists, heart biologists, to start looking into this. And the idea would be, can we use combinations of sound waves, electric fields, or new materials to improve that? So again,
We might need a completely new heart.
I don't think we can make a new heart. I don't think so. We probably hearts. I just the way right now our technology is beyond where I can see. I mean, a heart cell is such a clever thing, and as it has to fit so perfectly with each other, and as soon as it mismatches tinily, you have arrhythmias. How can we improve that? I don't think we can. We might be at some point when we know more about how we get the cells to regenerate themselves. I think it's very far.
One more question from me, and then I'll open up to the audience. You mentioned about traditional medicines, the ability that some people seem to have to intuit, as it were, what's wrong with people by feeling their pulse.
I don't think
The
they
mystery-
intuit, they measure.
So they're measuring. I mean, have you some ideas how things like acupuncture manage to do the wonderful...
We don't know.
Is it physics or is it biology?
It might be neither of them.
Might be psychology.
I agree. So one of the beautiful things of traditional medicine is that as you give yourself to another human, you're giving yourself to another human, you're giving your hand, your body is touching you, you are reconnecting yourself with your own body. Medicines don't cure you. Immunotherapy doesn't cure. It's your body heals when you help it heal. So I think many of these traditional medicines help us to reconnect our brain and the whole body. We become more aware. Things we don't understand. We know our brain is connected to the rest of the body. Obviously, acupuncture doesn't cure cancer, but for many people it's useful. How does it work? It's very complex because we are complex, and as I said, we're entangled with the environment. And as many doctors tell you, a lot of the cure has to do a lot with the physical and intellectual relationship with the doctor. Part of the process of curing is dealing with your doctor, that information flow, the physics, the emotions that go into it. Every doctor will tell you that. That's why I think AI will be good in the clinic for diagnostics, but I don't think we can substitute a nurse or your mother or your father helping you when you are ill. We're much more beautifully complex and all that. And I think traditional medicines go there, and that's why people look for traditional medicines. Because the reductionism we know is good, but it's not enough. And you need that other things that we still have no clue how they work. Again, I finished saying, what does the role of emotion play in our lives? We don't know.
Questions from the audience? Owen?
So I was very interested, well, brilliant, everything's brilliant, thank you very much. I was very interested specifically in the stuff about the role of electric signals, because many years ago I read a book called something like Electromagnetic Man, and it was basically about a researcher who had mapped the body from a magnetic point of view, and it sort of coincided with the Chinese medicine meridians.
I think, I'm not saying Chinese medicine is completely right. I think Chinese medicine is also a construct built after the revolution. It's a complicated thing. Even the field of traditional Chinese medicine, Mao creates it in the universities as something you can go and study. Before it had not been so. But it does bring a lot of tradition of understanding the body. And traditional Chinese medicine and other traditional medicines that look into the physics. That's the basis of Qigong, for example. You get meditation, you try to see the flow of your energy, and you get ill when your energy gets stuck. Okay, we don't know, and how can you do that? And we don't even know if the brain can map it. But I do recommend you to look at the work of Michael Levin in Boston. Okay, so he's revolutionizing biology, but reassessing the role of electric fields in embryo development. So, for example, he gets frogs and tadpoles and planarias, little worms, and messes with the electric signals between the brains and the body as they grow. And everything goes out of shape. They can grow two heads, they can grow an eye in the gut and things like that. So what he is establishing or re-establishing, because there was a lot of research in this before the genetics and the proteins and all, they took control of everything, that people saw that you can stop a tumor growth with an electric field. You can, electric fields are fundamental for the way cells feel with each other. It's not just genes, it's how the genes are expressed.
So I didn't actually ask my question, but you're kind of answering it, which is, we're surrounded by fields, and we've got health regulations that are based on models of how people think they may or may not interact with the body. But do you think that they need to be revised?
So one interesting piece of information for you is that in Russia the regulation is different. And one of the reasons was that during the Soviet Union, Russian biologists were not so obsessed with genes and proteins, and actually they continued doing physics. And they did a lot of experiments with humans and electric fields. And they have certain frequencies in Russia are not allowed because they think they interfere with human biology. Of course they do. We don't know how harmful or less harmful it is, or how resilient our body is. Remember, biology emerged on Earth in extremely harsh conditions of the early Earth. Before we had a good atmosphere, we were, bombarded by a lot of energy from space. We're pretty good at surviving radioactivity. We might be good at dealing with electric fields. I think we're pretty resilient to a lot of things, so I can't tell. I just tell you that the Soviet Union had different laws and that Russia still has that. And there was this weird incident in Cuba with this American embassy that people started having headaches. Remember that? Not that long ago? Do the Russians know something that we don't know? Probably they do.
So one of the people who speak at London Futurists from time to time, Andrew Vladimirov, who grew up, as he calls it, in the Soviet Union, although it was part of Ukraine at the time, he talks about Michael Persinger and other people who have done a lot of thinking about the electromagnetic nature of the brain and some of this. You have a question?
Yeah, no, it's related to that. Is there any study going on about this electric?
Yeah, big time. I tell you, Michael Levin is open the field. Because the moment he shows you can use electric fields from regenerative tissues, for perhaps dealing with tumors, for tissue engineering, we're going away from the part, which is great. Because also in biology now, there's not only people doing genes, there's people like me, engineers, and you hear it. And engineers are great because if it works, it works. We don't care how you do it. So that's why tissue engineering is so interesting, because it's a field usually doing biomedical engineering departments. So people will be very happy to put an electric field and see what goes on. And it's just, you just see it. All our cells are electrical, not just our brain, our hearts, lots of the fibroblasts have, all our cells have electrical properties. We are mechano-electrical chemical devices from the nanoscale up. Because we emerge from that. The Earth contained all that. We emerged from shaking, electric fields and radioactivity, and everything else was in the origin of life. So, of course.
Peter?
Also, the water molecule is a very interesting molecule because it has particular electric properties.
The water molecule is the biggest mystery, and we still don't know what is the water structure and the ice structure.
Yeah.
I just did some experiments in the lab. We're publishing next week. I'm interested in freezing. And I don't know if you know, water just below 5 degrees expands. So the water structure gets different at every temperature. So for example, we found one of the reasons probably cells break. That's why if you put a banana in the freezer and gets a mush, or that's why it's a big problem for egg freezing, which people don't tell you. But the success rate of getting pregnant out of a frozen egg of yourself is less than 5%. I think might be below 1%. And one of the reasons is that they just get damaged. And for example, what we saw in the lab, below 5 degrees, the water changes and expands the membrane. So the whole thing changes. I mean, as soon as you saw these nano machines that they're moving, as soon as you change the water structure, the whole thing goes different. We emerge from water. Again, going back to entanglement. We emerge from entanglement from our environment. Change the environment, we don't function. So one of the big things of evolution has been able to create resilient life to many different conditions. I think that's one of the reasons why we have so many proteins. Because when one system fails, when physics fails, another little nano machine kicks in, and another one, and that's why we can't get rid of cancer, because we kill it and the cancer cell does bang, 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 always comes back.
We are killing some cancers, aren't we?
We are.
The
But
immunotherapy you mentioned has...
And chemotherapies work also. So if you're quick and we're getting better, because we're getting better and we're getting humbler. Our understanding biology, I think.
Tony?
Well, thank you very much for such an inspiring, beautiful lecture. It was interdisciplinary, and what I really take it as most important for me is that you have given criticism to reductionism, but you also proposed something instead of it, or as an accompanying feature.
And we're not abandoning. I don't think we should abandon reductionism.
Yeah,
It's also in our evolutionary history. We should not forget our own history, and it works. In some things, it's great.
Yes. I have a dozen burning questions, but I only give one. and relates to new materials invention. And you mentioned that this is one of your most recent
Yeah, I love new
interests.
materials. I'm going back to my
Making the iron
stuff.
from wood.
Yeah, that's nice, huh? My
And my question relates to constructing a lift into space.
Oh well, we're going back to
We
the
are
lift
talking
into
about,
space.
graphene fibers.
Yeah, it was
So,
carbon nanotubes, right?
Yeah, the problem is with the weight and strength. And you mentioned the old.
Maybe we have to make it out of wood. Yes.
Yes.
I think so, because the problem with carbon nanotubes is that how on earth, is like my friend the sculptor said, give me graphene, so how on earth you construct something that makes sense out of this powder? It's powder. And what you want is to align the nanowires in one direction. We can do that in a small bits, but trees do it in meters. That's why I'm getting more and more obsessed with wood, because because we're learning also a lot of genetics. It's good, genetic. I'm not against genetics. It's just to deploy it with the rest, to put it in context. Can we make wood in shapes that we control better? Not totally, probably we won't. Better, and mix it with our materials to create the engineering of the future. I think more and more people are thinking that. China is putting a ton of money in research on cellulose. The US too. We need to grow them. I mean, even if you want to colonize Mars, if you want to go bonkers, you cannot bring all the steel from Earth.
We've got a few bonkers people here, by the way.
Yes, I can calm myself.
You had a question there? Yes?
Thank you so much for the talk. It's really inspiring as a hardcore reductionist. I'm kind of constantly challenged by new innovations. I'm really interested in what you described as instead of the sort of the nanobots or the proteins that we make, writing the genetic code and letting cells create the proteins, which is genius. I'm really interested in why the body clears
Nanoparticles.
that we
but it doesn't clear sort of proteins
Yeah.
that a cell
We still don't know that. It clears nanoparticles probably because we have evolved with a lot of nanoparticles in the environment. So the kidneys are made for that, the kidneys and the liver. One of the main problems of nanomedicines made of either DNA shapes of proteins is that we don't know how effective they're going to be. Again, they're being deployed in mice and cell cultures and they work, but we have no idea if that's going to work or not. That's why David Baker's virus is interesting. It's creating a bigger structure of something we know the body doesn't always know how to get rid of. A virus, again, is using evolution. Maybe we fail with our simple approaches, but use evolution, learn. So what he's proposing is to get the virus and let it evolve in the plate first. Play with evolution. I think one of the most, I don't know if it's going to happen or not, but I think at some point we're going to learn, or that will be the biggest human achievement, the algorithm that calculates evolution in the computer. Or it's not in a computer, it's in a mesh between computers and viruses in plates. That's unlimited power.
So to do that,
Just a
all you
minute,
need
so
is-
what about-
Maybe that's what we need for Mars colonization. Actually.
Mark?
No, I beg your pardon, I was just going
He's
to
been asking
I just
about
had a
it.
water comment, not to worry about. Oh, have you heard about Gerard Pollack from Washington State University
I have.
and his
I don't
research
know.
on water?
No.
He's got something called easy water. He's
I don't know what that is.
shown a different state of water, especially when it becomes ice.
Oh yeah. There's a lot of research into the different stages of water when you become ice, because we just don't understand how they're different ice structures.
Okay, so he may shed some light on that for you.
Okay. I'll have a look at that.
Wonderful.
Yeah.
I had just a little question, and thank you so much for that. It was so interesting and inspiring. Thank you so much for that. At the beginning of the talk, you mentioned about how we are discovering more order and structure in the smallest parts of nature, and we're finding like more cosmos and less chaos. I wonder
I
if it's
didn't say that.
well, kind of that, like instead of understanding things are chaotic, like we are finding the structure and the order.
That's what the obsession of humans has been from the beginning, looking at a structure. Looking at a structure is the obsession of aesthetics, of religion, of building, of construction. This has been the human obsession, the animal obsession. This is very old. This is the yin and yang, right? So
Yeah.
this is in one of these dualities of nature. We crave for order and structure, and yet emerges from chaos, right? And so what we are finding again is this dialectic. We wanted to forget the dialectic. We could just reduce everything to something we could control. And nature is coming back to us to tell us, you're out.
Okay.
So we're rediscovering the dialectic of complexity and simplicity that has been the root of human creativity, religion, arts, of everything else from the beginning of humans. So it's a great time.
So actually my question will be kind of like, is it like, actually I think you answered partially that both concepts are integrated in the,
Yeah, we see it all the
in the
time.
reality.
We
rather
see it
than
in the way we understand, in the way we construct, in the fabric of reality as far as we can perceive reality. These are epistemological questions. I'm a physicist, and in physics we accept everything, even quantum mechanics. Yes, we are reassessing the nature of reality and the way we learn. And the beautiful things about machine learning and artificial intelligence is it gives us a new tool to understand all this.
So, Peter, I'll let you back in again.
Oh yeah, I was just wondering what you thought about Craig Vinters' work with synthetic biology and how that might tie in with your work.
I don't follow him so much anymore. I don't know what he's doing now. Synthetic biology is everything I told you now. Although synthetic biology, as everything is in academia, starts getting in these niches and people not collaborating with each other, has become just part of what is the greatest possibility of synthetic biology. Again, it's always with the funding, and there's a lot of wonderful work in trying to get bacteria to produce energy, in trying to get bacteria to produce glues, or I know these people trying to get bacteria to that so that concrete heals.
Yeah.
They put it in
Yeah.
concrete. It's part of all this revolution. Synthetic biology is about, again, building with nature.
Yeah.
And learning how nature works, because on the way we're learning how much, of course there's always been the obsession with DNA. It's
Yeah.
this obsessive going back again to the molecule of life. How much DNA can you cut? How much DNA can you not cut?
Watson and Crick sold it really well.
Which one?
Watson and Crick sold it really well.
It's beyond, I think it's before. I think the obsession with the genes is
Francis
offers
Golden.
is measures humans, and it offers, and it came together with Darwinism, social Darwinism. I learned recently, and this was surprising, and yet I see that one of the reasons why in the US there's so many creationists is because people were going and telling them, you're poor because you're inferior. And I'm not poor because I'm inferior, you know that. It's the conditions make you. It's your entanglement with the economic environment that is making you. I'd rather get God to make me poor than you and your superiority. Probably you've read Superior by Angela Saini, or seen the television program she's made about the origin of genetics and eugenics in the UK.
Who's the person?
Angela Saini.
Yeah,
she did a terrific program about
and the book is wonderful, and it shows the history of genetics, and actually for me the surprise, I knew there was obsession with genetics for colonialist reasons, for patriarchy reasons. And this is also in Angela Saini's book, when the Europeans, the British, arrive in Australia and they see the Australians that they don't need to do agriculture to survive, because they live very entangled with nature, they understand how to get food from nature. Instead of saying, wow, this is great, they say, oh, these people are inferior because they don't have technology. I think this idea that the capacity to have a reductionist technology that uses nature and modifies nature, and then you feel superior, and immediately you need a cultural framework that supports that interpretation. You need to prove, because I think you're never convinced.
Well, that's what Stephen Jay Gould wrote about in
Correct.
This Measure of Man.
It's
He-
exactly the same story.
Many attempts by white male scientists to find measurements that prove that white male scientists were the
Are the
smartest
best.
people on the planet.
And again, I mean, and why you do that is the same thing to prove that women are bad at science. And I've been told, still tell me, you're not good at physics, you're a woman, you're good at languages. I'm good at both. It's this idea that you can objectively find a route to that classification that puts you at the top. That you need it. This is a psychological thing of humans, that you want to prove you're better, and when even though you are better already because you have more power, that doesn't seem to be enough. And then the genes are perfect. And the statistics. This another obsession of biologists is never look for causation. This process of trying to understand reality of where it comes from, but correlation. The statistics and correlation is the key to biology. And indeed, I mean.
I see lots of hands. I'm going to try and take a few more points. Rohit?
Thank you. I missed the beginning of your presentation, but I really enjoyed the part I have heard. I'm left wondering about the challenge for society. That on the one hand, we have incredibly talented scientists like yourself, really pushing the boundaries, and able to communicate it to interested audiences like the one in this room. But then we have, if you look at our election at the moment,
and the scientific community itself.
You talk about duality and reductionist. The whole way that conversation about science is going on is simply that it will create the next generation of industries and jobs.
I'm not sure. I'm giving you where I think is. I'm not sure. I'm giving you what is our best chance of improving technology. At the same time, there's a lot of people within the scientific and economic and everything else, all the relationships that create scientific discovery that are not working in this way at all. It's very hard for me or any of us to get any funding. Progress is unclear. I give what is possible, what I think is the best scenario I'm trying to create a vision of a positive future where we survive, because I'm not positive myself. Okay. And again, going back to your politicians and reductionism and complexity, we have a huge drive for destroying the very fabric of our society at the moment.
So my question, and fascinating to hear what you just said, is a lot of the stuff we're talking about, in order for it to get embedded in society, would require fundamental shifts. If we're going to live longer,
shifts.
If I just finish, if we're going to live longer, that means transformation of pension systems, rethinking jobs, all sorts of things. How do we start to really communicate the power and the potential of what people like yourself are working on, not just to politicians, but to broader society and the media, so that they truly understand the game-changing nature of the work that's being done, even if it's not all being funded, and even if it's not being done the way you want. What we can see is that there is a big shift coming in the kind of scientific breakthroughs that are happening. How do we raise the understanding of that so we can have meaningful dialogue?
Yes, so I don't think scientists should drive it. I don't think anybody should drive it. The future has to become a dialogue ideally between different people. I'm quite obsessed of how there's this idea of the sequence of the technological revolutions, and they usually they're talking about generations, technologies, overcoming technologies, and I think one key question that never gets researched is the creation of culture, and so that's why I talk about art in the end. I think we need.
Give away a spoiler to your book. They say that in the end, there's a lot about the interplay between science, technology on the one hand, and
the arts and society. Right. I think not that my science colleagues care, most of them, but
some of them do. Don't
some
they?
of them do. Correct. So by creating alliances with people, by talking, creating dialogues, trying to transform culture, one beautiful thing that is happening is there's more artists interested in technology, trying to work with scientists to understand. I mean, most of it is rather crap, but some people are really doing big efforts to create cultural transformations that allow this kind of dialogues. I think we need to we're going to have a massive struggle in the coming years for keeping the line of progress of humanity. And part of me writing the book and talking to a lot of people is like, can we identify ways forward? I talk a lot to politicians, to people. I just got invited, I think, to the European Union Parliament to talk about these things. We need alliances among people that want to transform things. I will talk to the politician, and together we need to transform the cultures and try to have a plan. We need a plan for when the things start collapsing. They don't look great. We need to start constructing plans. It's very complicated, and I agree. There's hope. There's a lot of people starting to think, I think, in the right direction.
As well as being invited to the EU, aren't you on Radio 4 start of the week?
I am on the start of the week. Yeah, whatever that will come up. Yeah. I think in a way, we need a kind of new Bauhaus type of idea. If you think the Bauhaus collapsed after the 19...
This is an architectural school.
Not only architectural. So after the First World War, Europe was a total wreck, shocked and traumatized. Architects and scientists, and not many scientists, architects and artists got together in a sort of the vision of the Bauhaus, create technology that lives with nature, bring together, that was their idea, bring together the crafts, the arts, to create an environment where we can live and survive. It didn't last long. Hitler and the Nazi party got rid of them pretty quickly. They had other reductionist plans to get rid of but maybe we need a new kind of Bauhaus here. You kind of thinking together, bringing the arts and the technologies to create new cultures. The Bauhaus here now is everywhere. It's in IKEA and in your iPhones. They revolutionized design. They created design. We need something. We don't know where it is.
Now, you've been part of the Future of Humanity Institute in Oxford.
No, I've never been.
All right.
I was director of an Institute of Nanoscience for Medicine, but in the Oxford Martin School.
In the Oxford Martin School.
Yeah, but I've never been part of it.
But wasn't the part of the vision of James Martin?
He was.
To have that same kind of bringing together of people to look at
the problem is the mess and the sort of the crisis in capitalism and the crisis and everything else makes it very difficult for people to come together. We are disorientated. We are bombarded with bad visions of technology. One of the reasons I wrote this book is because most of the books people are reading about technology are not written by scientists, and they're not written by women or by working class women from Spain. Yeah, I don't know. It's complicated.
Rupesh?
Yeah, just sort of going back to the therapies, rather than trying to reduce solving the medical condition, specifically trying to work out how it works, just kind of like throw nature at it, and as long as the outcome is as desired, you don't need to know how it works. But I just wonder if you're taking into account sort of
I think we will want to know how it works, and I don't think the current I think the Nobel Prize for the person that figured out how aspirin worked, it was as late as the 1990s.
Sure.
So we've always worked a bit like that, right?
But just the idea of letting nature have a go at it and then figuring it out afterwards. I mean, if you're taking into account evolution...
I'm not saying that.
But it's sort of more like rather than trying to work it out,
we are trying to work it out.
But after the fact that you found a therapy that works.
I don't say that. What you said is I'm trying to say that all the information we have accumulated in genetics, in proteomics, the new methods of mathematics, of physics, allows us to have more than just have a go. We're not doing trial and error. The beauty of the science that we can start deploying from now on is that we can work with nature in a much more informed way than we've done in the past. What I'm not saying is I'm not at all talking about trial and error.
Sure, but the idea of a holistic therapy rather than a specific therapy.
Yeah. And
So, if you're thinking about this holistic way of looking at it and taking into account the evolution of these nano machines,
I'm not talking about evolution of nanomachines. No, I'm talking about evolution in biology.
evolution in biology.
Yeah.
Sure. And the thing is, though, what I'm thinking about is that there are sort of like some conditions that are, I'm thinking specifically about sickle cell, which tends to affect Afro-Caribbean people more. And if you were to use this kind of technology to deal with sickle cell, would it then lead to the possibility of having like ethnically sort of targeted therapies? And if a lot of this research is going on in East Asia, and you mentioned that a lot of testing is done on male lab rats, is it possible then that you will have therapies directed at certain demographic groups? And they work, but it could be that you have a therapy that works well for Asian people, say East Asians, and you have a therapy that works well with Afro-Caribbeans.
The problem is that race is much more complicated than our definition of race. You cannot find biological base for race. So no. People may try, and people probably do. It's important to do research on all socioeconomic and ethnic groups in society and involving everybody because exactly of that. Because we come from a very complex interplay of biology, economics, social background. I don't want to reduce research to race or gender because that's not enough.
Well, I just wonder then, because it's like East Asians don't process alcohol as well as Europeans.
Many East Asians, some do.
Yeah, and similarly like the sickle cell example.
The sickle cell, yeah, it's a statistics. This is the complex interplay between, but I mean, find the roots of the disease and try to find a cure wherever it comes from.
I mean, potentially going forward.
What you're talking about is what we call personalized medicine.
Yeah,
we're all different.
Right.
We're all different, and we all respond differently to drugs and to everything else because the way we grew up, the way our bodies grew up, where we are, where we come from affects the way you do it. So for most people, aspirin works, some people don't work. The way we do now medicine is that we produce drugs that work for an acceptable percentage of the population and is not toxic. And then you can put it in the market. It's not killing people and works for a number of people. But the dream is personalized medicine, which is going back to my picture as good biosensors. Remember the pulse, but the clever pulse. Good biosensors that test you as an individual in that point to design your therapy. That's the dream. And it will not only be genetics and proteomics, it might be a combination of other things. This is what we're trying to figure out if we can, and this is what I was trying to tell you. We need to put together everything because we're complicated.
So you're almost out of time, but there was still one or two hands I saw. Do you have a question, Terry?
Yeah, thanks. That was a very optimistic.
No, it isn't. This is what I am. I'm not. I think it's our duty to create positive visions. We need to have a plan. I'm not optimistic.
Well,
at all.
It's interesting you said that. You did qualify that at the end in your comments just now.
I think being optimistic is a duty for survival.
Absolutely right.
Another thing is what I think.
But, I guess
my intuition is not so clear in this case.
I mean, it seems to me that people like oil companies and so on can be easily dealt with because that's simply economics. Oil companies, you're talking about survival and so on. Oil companies, drug companies, anything in the commercial sector can be easily dealt with because that's a matter of economics.
That would come as a surprise to Ralph Nader.
But it seems to me that the thing that is not easily dealt with is things that are essentially anti-science, like the Green parties, for example, who you would think were concerned with survival, in actual fact have an ideology which is an ideology like other people's, a destructive one, which is, for example, preventing a lot of science being done in Europe.
Yeah, but again, this is because of the history of the deployment of science on societies. And again, I think it comes a lot from the history, particular history of the English-speaking countries, which dominate so much the history of what the media tells you. So the Industrial Revolution in England create scars forever in the psychology of people in this country and in any English-speaking country. But I'm from Spain, and we're much more positive about technology. And if you go to Japan or Asia, because their economic development and their well-being has been always tangled from the beginning with the technological increase in the country. Japan well-being as a nation came with technological development. It's difficult to find a Japanese person that is very negative about technology.
Yeah.
They worry about their environmental aspects because it's rooted in Japanese culture, the relation with nature. And many, indeed, Japanese artists have always found something which I find very beautiful, that they realize that a key of technology is human communication with each other and with nature. And technology, one of the main uses of technology is to connect ourselves with nature through science. And so, yes, it's very complex and it's very cultural dependent. And many, even many scientists, I'm just talking about Watson, right, who has been, as the Watson and Crick, who just been deprived of his honorary degrees all over the world because he just is a racist that uses genetics to see he's superior. Science is entangled with the cultures where it breeds, and within the scientific communities, there's all sorts of tendencies as well. I'm putting mine forward.
So let's try to finish on an optimistic note, because you did say it is our responsibility to try to be optimistic.
Yes.
So where can we find grounds for optimism? If things are going badly in Britain and America, should we move to Spain or
No,
Japan?
Spain is doing as bad. And Japan is in a climate of cultural, economic, and everything, a stagnation with the same party in government almost from the Second World War. And increasing racism. No, it's complicated at the moment, which maybe is good. I think maybe we will only change and we will move forward when we really get close to.
So you're saying it has to get worse before it's going to get better?
I
could say something optimistic.
Yes,
please do.
The younger generation.
I agree.
The younger
They're
generation
wonderful.
are much more
aware.
Climate change. They're much more interested in collaboration to make a better society. I mean, basically
all over the world, and it's not only Europe. I teach sometimes a course in Sichuan University in China, and I always come back really happy. The obsession with the environment of Chinese students, the interest in gender equality of Chinese students, the or indeed my students at Oxford, or everywhere I go.
Latin America as well.
Everywhere. Latin America, again, is in a total social...
Even the States.
The States in particular. I find the students from the States, because they have such harsh realities that they get when they go to university, the lack of everything, that they're very aware. Actually, perhaps the American students are the ones that are more aware of the horribleness of the situation. So there is a lot of hope, because they understand very well that the States are, is their future.
It actually reminds me of the 60s, because, there really was an enormous amount of optimism. It surrounded war rather than some of these other things, but there, that's where it started in the States anyway, was with the protests.
And I don't think,
against the
I'm not an expert in social revolutions, but you don't need everybody to agree on a social revolution for the social revolution to happen.
Right.
And that's what I think you need to put good stories forward from alliances, and try to create a group of people that can lead to the next transformation, and cultural transformation, economic transformation. And as we know from physics, phase transitions can be very fast. We are now in a phase transition that is gone quite fast. And maybe we also need to apply physics and complexity to our understanding of societies and how we transform societies. So again, being a scientist and finding the right social scientists to create the right networks that brings positive change is the task we need to do.
In some of these protest movements and activist movements, such as Extinction Rebellion, they explicitly study how they think positive social change can take place. So maybe then we shouldn't have just a single-minded focus on just the younger generation. Maybe there will be a diverse approach, a multidisciplinary age approach.
I'm not a Confucianist, but Confucianism has a good thing,
Yes.
which is that you care about the next generation. And I think making links between generations is very important.
So I'll ask you to thank Professor Contera in a minute. I just want to say that London Futurists continues in 5 weeks' time. Somewhere in this building, we will be looking at some stories about the future. Stories about how the future might be if there is more automation, which is threatening our work and threatening our value. What kind of future might that then be? And we'll have John Danaher, who is coming from Galway in Ireland, to talk about his thesis, which is that actually we shouldn't get too excited about the prospects of humans and biology merging. Sorry, humans and technology merging. That cyborg approach probably isn't the right way, and he's going to point to other possible
A
futures.
little bit cyborg is good, no?
A little bit of, I mean, he is a fan of being a cyborg, but he says we need to go beyond that. So that's in 5 weeks' time. Some of us might go to the pub next, and we can have a chance to look at Tony's 11 questions that he was very polite not to ask. He gets the star for just asking one question, so I like that. And maybe some of us can discuss it in the pub. But thank you so much, Professor Contera. This was really a very special talk.